Welcome. This is going to be for lab number 8, urinary system and urinalysis. This will include material from exercises 29 and 30 of your lab exercise book. So let's get started. Now the objectives include review the gross anatomy of the urinary system, including any models and/or dissection models. Some of that may be seen in the book or in some of the videos that I do include for this particular lab. Review the microanatomy of the nephron, renal blood supply, ureters, and bladder, including review the slides and the micrograph. Review and examine the physical and chemical characteristics of urine and relate the function to the urine nephron structure. Now, in this lab, we are not going to have hands-on with doing some of the analytical exercises, but I have attached into the lab video file several lab YouTube videos, I should say, that you are absolutely essential to view. They will give you a perspective of various factors and various means to do urinalysis. Therefore, your worksheet will include pages 535 to 536 and 547 to 548. These are attached and you are required to fill them out and upload them before Friday, 5 p.m. Let's get started. When we examine the gross anatomy of the urinary system, we note the presence of the adrenal glands loosely attached above the kidneys. And I put above because the other way to call the adrenal glands is suprarenal glands. Now, the kidney itself, its gross anatomical structures are the hilum, which is an indentation of the kidney surface where renal artery, renal veins, and nerves enter. The ureters exit the kidneys. They are the conduction tubes for urine. The kidney structures include the renal cortex, which is the outer region, and the renal medulla, which is the sort of middle region that contains the renal pyramids and the renal columns. Nephrons will occupy both the cortex and medulla. There are two types of nephrons, the cortical nephron and the juxtamedullary nephron. In the structure within the kidney, you have the renal pyramids. These are cone-shaped structures of renal medulla that contain nephrons. The renal papillae is the apex of the renal pyramids. The renal column is sort of this inward extension of the cortical tissue that separates neighboring renal pyramids. The kidney lobe contains various renal pyramids and adjacent renal cortex and renal columns. We have two calyxes here. You want to keep clear on the separation of each and the understanding of each. The major calyx is the junction area of collection from the minor calyx. The minor calyx is a collection site of urine from the renal papilla. The renal pelvis is sort of this funnel-shaped structure that's continuous with the ureters. Then we have to deal with the urinary bladder, which is the collection structure of urine. Now, there are two sphincters that control urine release. The internal urethral sphincter, which is a thick band of smooth muscle. Keep in mind, smooth muscle under autonomic control. The external urethral sphincter is a band of skeletal muscle. And you can rethink that as saying, oh yeah, because that's the one that we have to learn as we're little kids. It's basically the voluntary structures, the voluntary control, I should say, that we learn to control when we void our bladder. Now, if we go to the bladder, we notice some structures internally. The trigon, which is a triangular base of the bladder, it's marked by the two ureter connections and the urethra to the bladder wall. Rugae are folds in the bladder wall. Upon filling with urine, the rugae disappear due to the expansion of the bladder wall. The epithelium in the bladder is composed of transitional epithelium. You'll also want to note differences in the location and structure in both male and females. In the urinary bladder, males, their bladder is superior to the prostate gland Females, it's anterior and inferior to the uterus and anterior to the vagina. The urethra is a muscular tube that conveys urine from the bladder to the outside of the body. Males have a relatively long urethra, which consists of prostatic urethra, which passes through the prostate gland, the membranous urethra, which passes through the urogenital diaphragm, which is the muscle wall, and then the spongy urethra, which travels through the shaft of the penis. Females have a relatively short urethra, which travels anteriorly to the vagina and posterior to the clitoris. As you can see here, you have, of course, the diaphragm, you have the left and the right adrenal glands, you have the left and the right kidneys. Notice how the blood flow comes in. And you have this major amount of blood that gets shunted over from the descending aorta into the kidney, and then, of course, it returns to the inferior vena cava. You have to have a lot of filtering that occurs. Note that out of this hilum, you will have, of course, the ureters. They will eventually come and meet in the urinary bladder. Okay? You notice also in this structure, and I would encourage you to review this diagram, you have, of course, the ureter, the renal pelvis, the renal papillae. You go through all of the structures. You have the outside fibrous capsule. You have basically these fibrous capsules within the renal sinus. You have the renal cortex here, the renal medulla. This is what is sort of the renal pyramid here, with the renal papilla down here. You have a renal column structure right here, and of course, you have this entire section here, which consists of a renal lobe. Now, when you look at a comparison between males and females, you can see where the ureter comes out and goes in to the urinary bladder. You notice the location of the urinary bladder. And you can see this in the female, it is superior to inferior to the uterus. The bladder here is superior to the prostate. And here, of course, you have the urethra. Notice the urethra is much shorter here in females than in males. Now, when we go through the bladder, we have several important structures that act as support ligaments, the middle umbilical ligament and the lateral umbilical ligament. There's a big, thick area of smooth muscle here. This one on the top here is called the detrusor muscle. Now, notice in here these different colorations. This is really rugae. This is the folds that occur on the internal surface when the bladder is not filled. They sort of disappear as you expand the bladder as it fills up with urine. This is the uretic orifices as well as the trigon. This is the neck of the urinary bladder, and that contains the muscular internal urethral sphincter. The external urethral sphincter is lower, and in males, it would be inferior to the prostate. Okay? Now, when we examine the nephron, the functions of the nephron include, of course, glomerular filtration, which, of course, this is where we deal with the filtration of water, glucose, amino acids, electrolytes, nitrogen-containing waste pass from the blood to the nephron via pressure gradient. Okay, so all this is occurring in that glomerulus. We also have tubular reabsorption. This is where we begin to pull back on certain materials from this filtrate, and that's usually water, glucose, amino acids, and some ions, and they return to the blood. But we also finally have tubular secretion. This is the basically re-dumping back into the filtrate excess potassium and hydrogen ions, drugs, creatine, metabolic acids transported from the capillaries to the tubules. When we talk about structures, we have to talk about the glomerular capsule, the glomerulus, which is the tuft of capillaries inside of the glomerular capsule, and the renal corpuscle, which consists of the glomerular capsule and the glomerulus. As I said to you earlier, the term filtrate, this is fluid entering the glomerular capsule. This is basically considered raw urine. But we're going to go through a lot of processing because when you think about it, you basically each day filter out, make raw urine, 180 liters, and yet it's only about 1.8 liters that we excrete every day. That's because of the variations that we have in pulling back water, glucose, some of the ions, etc. Now, the components of nephron include the proximal convoluted tubule. It will be referred to PCT. The nephron loop, which is also referred to as the loop of Henle, and it's a descending loop and an ascending loop. And then the distal convoluted tubule, which is also referred to as DCT. The connecting tubule, and then the connecting system, which comprises collecting ducts and papillary ducts. The papillary duct, this is where urine from several collecting ducts empties into this duct. Now, as you can see, here is this magnification. Here is our nephron. You can see here the glomerulus. You have the proximal convoluted tubule. Then you have the nephron loop, descending, ascending. And then this longer distal convoluted tubule. And then, of course, it empties up into the collecting ducts. Now, this is a cortical nephron. How do you tell? Because 85% of the nephrons are in the form of a cortical nephron. What you have here are the juxtamedullary nephrons. Think about it this way, crossing deeply into the renal medulla. And it is in these ones that, yes, you still have glomerulus, you still have PCT, but you'll notice that you have a much longer nephron loop of the juxtamedullary nephron, much longer ascending loop, and then of course you have your DCT here with it emptying into the collecting tubule. This model gives you an idea of some of these points, and I encourage you to review some of these images. You can see the glomerular capsule. If you go in, you will see the glomerulus. Okay? And you will note also the arrangement, the architecture of the blood vessels that go from basically the interlobular to eventually you will get into some of the smaller amounts. Keep in mind you also have the arcuate artery and vein here. Okay? Now, when we study the blood supply to the kidney, it consists of the renal artery, which enters the kidney at the hilum. It then breaks into, or branches into, segmental arteries, then interlobular arteries. These branches travel toward the cortex through the renal columns. Then the arcuate arteries, which are branched from the interlobular arteries. They are the landmark for corticomedullary boundary. Then we have the afferent arterioles, and these are the actual arterioles delivering blood to the glomeruli. Then we have the cortical radiate arteries, which branch off from the arcuate arteries and ascend into the cortex. From there, after you've left the glomeruli, you have efferent arterioles. The blood is exiting the glomeruli. It has lost some of its product. You have peritubular capillaries and the vasa recta, which arise from the efferent arterioles. The peritubular capillaries, these are closely associated with the renal tubules in the cortex and the nephron loops of the cortical nephrons. The vasa recta surround the nephron loops of the juxtamedullary nephrons. The peritubular capillaries and the vasa recta form this complex blood vessel network around the kidney tubules. Now, capillaries from the nephron tubules drain into the venous system that drains blood from the kidney. So from basically the capillaries, we go to the cortical radiate veins, the arcuate vein, the interlobular veins, the segmental veins, and the renal vein. As you can follow through in this diagram here, renal artery, segmental artery, interlobular artery, arcuate artery, cortical radiate, afferent arterioles, glomerulus, and of course then you have the efferent arterioles and eventually the cortical radiate veins, arcuate veins, interlobular veins, renal vein. Very nice. A couple of other points to keep in mind. Remember that I said that the cortical nephron and the juxtamedullary are a little bit different in the sense of what you see here, and that's the length. Once you have blood leave from the glomerulus, you have it going from the efferent arteriole, and then it starts forming these peritubular capillaries, which kind of wrap around the kidney loop, or the nephron loop of Henle. And eventually they leave, and as they do, the blood will enter into the cortical radiate vein. Remember, peritubular capillaries are taking away some of the material that we had by reuptake from the actual filtrate. Now, when you look at the juxtamedullary, much more complex. When you leave the glomerulus, you have the efferent arteriole, which kind of wraps around a little bit here, the peritubular capillaries here. But then they start going down, they go in these very long channels, which eventually end up being more vein-like due to oxygen loss. And what's going on here is these are what the capillaries of the vasa recta are. These are going to basically take up the reabsorption of various water, glucose, and ions, etc. When you examine the microscopic structure of the urinary system, unfortunately, we don't have slides in this particular exercise. What we do have are some micro images, and I encourage you to spend some time with those in the lab text. You will note renal corpuscles, glomerulus, capsular space, the epithelium, the renal tubules, collecting ducts, ureter, mucous membrane, which is known as the mucosa. Now, this is when we get to the ureters. Why? Because what we're going to have to do is that when the fluid comes in from the kidney, it doesn't just kind of drain by gravity. It's helped along by peristalsis. And for peristalsis to occur, you have to have a layer of smooth muscle. That's the muscularis you have. So you have transitional epithelium, laminar propria, smooth muscle layer, the adventitia. When you get to the bladder, you'll note differences, if possible, filled versus empty. I have not seen slides that really provide a good example of the filled. The mucosa will be a transitional epithelium, and the detrusor muscle, which is referred to as the muscularis. Here in these micrographs, you can see the distal convoluted tubule, what is called the Bowman's space. This is the glomerular capsule, which they've also referred to as Bowman's capsule. It is in this space that whatever the glomerulus filtrates out, this is where the raw urine is going to show up, okay? Now, when they talk about the vascular pole, this is basically the attachment where the blood vessels are coming in, the afferent ones, and then the leaving of the efferent. You have the proximal convoluted tubule here, the distal convoluted tubule here. Just one thing to help you, though, is that if you look at these cells, they're what? Cuboidal. They're simple cuboidal epithelium. Okay, you can see nice round nuclei, and you're forming this duct tube to take away or process the urine. In the nephron loops, you will notice various developments on the edge of the lumen. You have the descending thin limbs. You have the thick ascending limbs. These are structures that are involved with processing of and reabsorbing of those materials, such as water, glucose, sodium, potassium, etc. Okay? Now, if you get to the ureter, you'll see that, okay, there's a nice grouping in here. There's a laminal propria here. There's a transitional epithelium. And yes, even here, we're not talking about just the bladder now, because the tube has to expand a little bit as fluid is passing down through it. Then we have a layer of smooth muscle here, which it will contract and push fluid down toward the bladder. When we get to the bladder, we notice that the mucosa here is consisting of, again, transitional epithelium, a lamina propria, a submucosa, and then the detrusor muscle. This is all smooth muscle that will help play a role in squeezing, contracting, and eventually forcing urine out into the urethra. And you can see the structure here. Now, students here are not going to be doing the dissection of the kidney. If you were, you would be handling the scalpel with care. You would be taking and looking at the external anatomy, the renal capsule, hilum, renal artery, and renal vein and ureter. I've also tried to get a video to include a basic kidney dissection. When you make a slice, sort of a sagittal slice through the organ itself, you will get the internal anatomy of the renal artery and vein, segmental artery. The problem with some of the other structures is that it gets sometimes difficult dependent on what is the supplier of that particular kidney. Is it beef or is it sheep? And how long has it been since it's been perfused, etc. But you are encouraged to try to identify this from your diagrams and charts. Here would be an example of a right kidney, the frontal section. You can see definitely here the renal pyramids, the renal columns, okay, the minor calyx, the major calyx, and where it all empties into is into the renal pelvis and then down into the ureter, okay? When we correlate nephron structure with urine production, as we mentioned before, three basic functions: filtration, reabsorption, secretion. In filtration, the renal corpuscle, the glomerulus and glomerular capsule. You have the filtrate, which passes into the capsular space only if the pressure is greater in the capillaries than the capsule. Considerations: you can't have blood proteins, only ions, water, glucose, amino acids, nitrogenous waste. When you start showing up with blood proteins, there's something radically wrong with the basic healthy filtration that one would be seeing, because blood proteins should not be showing up in the urine. And if you remember that there's a formula which basically is composed of the glomerular hydrostatic pressure, GHP, which is the pressure of the blood in the glomerular capillaries. The pressure tends to push water and solutes out of the blood into the filtrate. You have also the capsular hydrostatic pressure, CSHP, which is the resistance of fluid in the capsule to allow more fluid entry. The blood colloid osmotic pressure, BCOP, that's the pressure of fluid to return due to the colloids in the blood. Remember that blood is not only having formed elements like red blood cells, white blood cells, as well as the platelets, but it's also going to have a large number of different types of blood proteins. These are going to have an effect of trying to draw water back into the blood vessel, okay? So both forces of CSHP and BCOP push fluid back into the glomerular capillary and thus oppose GHP. Therefore, we can have a formula. Net filtration pressure, NFP, force that moves fluid across the glomerular capillaries. So NFP is equal to GHP minus BCOP and CSHP. Now, if that got stunning for you, think about it this way. The net filtration pressure, that pressure to drive water across from the glomerular capillaries into that glomerular space, has to be greater than the blood colloid osmotic pressure and the capsular hydrostatic pressure. CSHP, think about it this way. If you had a plugged nephron, and at the proximal convoluted tube, let's say there was some sort of blockage right there. So fluid has filled up into the capsule of the glomerulus. That's going to be a strong CSHP. But most of the time you get very little. It's not a lot. What you do have situations where someone is very dehydrated. Therefore, their fluid capability or the fluid content in the blood, despite pressure, may be less, and therefore you're going to get less and less of the kidneys being able to filter out materials like water. Now, after filtration, we talk about reabsorption, which occurs through the tubule, but mostly in the proximal convoluted tubule and the nephron loop. Approximately 99% of water, amino acids, glucose, and ions are reabsorbed back into the blood. Secretion takes place throughout the entire nephron, but mostly in the DCT, distal convoluted tubule, and the collecting duct. This is where metabolic wastes, drugs, excess ions such as hydrogen and potassium are removed from the blood and enter the renal tubules. As you can see here, we have the juxtaglomerular complex, which includes the DCT right here. There's a little also patch of cells called the extraglomerular mesangial cells. We also have a little patch here of juxtaglomerular cells right here. And then we have this patch of cells here, macula densa, which monitor pressure and may be stimulated to release renin in response to changes in blood pressure and blood content. The afferent arteriole, as you can see with some of the peritubular capillaries, and we move into this area, we have filtration occurring through this through this glomerulus. And you have the visceral layer right here. And then when the fluid has gone through this, and we've lost some of the volume, therefore some of the, we've changed some of the viscosity. We have blood leaving by the efferent arteriole. This is the capsular space. This is the initial segment of the renal tubule. Notice that it has other factors in here, including some cilia. We've already talked about GHP and the other factors involved in creating a net filtration pressure. Keep in mind that the peritubular capillaries are absolutely essential with the vasa recta in being able to do some not only reabsorption, but to a degree, a little bit of secretion. Now, moving from the microanatomy, we're going to move into urinalysis. In all cases, even if we were back in the lab, we would not be doing this with actual urine because there's a biohazard. So the way they do it is they have synthetic urine, okay? And what you would do is normally you would note the physical characteristics of the urine. Okay, is there any cloudiness? What is the color? For example, darker the yellow, the less water is in it. Normal color of urine is about clear. Remember that urochrome is a pigment produced when hemoglobin is broken down and provides the yellow color. Turbidity is sort of a cloudiness, and this is due to crystals, bacteria, and dead cells. If you have an odor to urine, normally if it's fresh, is supposed to be sort of aromatic. If left out, you'll start having breakdown of the urea and you will have ammonia appearance. So it'll have that foul ammonia smell. You can also have certain other drugs, vitamins, foods, for example, coffee, affect the odor. Specific gravity. This is the weight comparison between a given sample of volume and an equal volume of water. The idea being that with a urinometer, which is basically a hydrometer of urine, the normal specific gravity for water is 1.003 to 1.030. Okay? Anything greater than that, you have indications of something not working properly with the kidneys. Here is the normal setup. With your sample, with the urinometer, you have it first done with distilled water. Then you compare that measurement with the sample of urine. Now, specific gravity is very, very simple. It's basically the density of your sample compared to the density of water. If the density of water is basically 1.0 grams per milliliter, then you should have something very close, very, very just minor difference for urine. This is one of those few unitless quantities of chemistry. So it's always measured in numbers, but not units. So what happens is that this has a weight, has a float, and so you're going to have the numbers wherever the final level of the liquid is. If the liquid comes to, let's say, this line right here, you have a specific gravity of 1.010. Okay? Now, examining the chemical characteristics of urine. Urine can be used to detect a variety of diseases and infections. Rather than have you go over and review the table of 30.2 for the list of testing items and associated pathologies, I would like you to look at that, but also I've included in the video collection for the lab, you are to go through at least the first few of them because those videos are going to be doing exactly some basic tests as if you were doing it in the lab. They're going to also do the Dipstick Test. The Dipstick is a fantastic tool. And I know you're sitting there wondering, get a life. No, I'm serious. Think about this. In a matter of a couple of minutes, by dipping that stick in, you have a bunch of different chemical tests. Now you have to be, of course, wearing gloves, and you also have to be savvy about knowing how to read it compared to the indication colors on the bottle. But if you do it right, and I had seen some colleagues who were teaching students how to use this in an actual class, or I should say for their jobs, you have to be savvy. You have to be able to read it quickly, make the decision, keep moving upward, and rotating the bottle until you get to the next test, and to the next one, to the next one. How quickly are you supposed to be measuring, and how quickly are the times? 30 seconds, 40 seconds, 40 seconds, 30 seconds, 20. I think it goes to like 50 seconds, and then 1 minute, 1.5 minute. Now, here's part of what I'm trying to emphasize. If you wait too long, the chemical reactions continue and you get an error in your reading. And that could be serious, especially if you're trying to make a determination of whether someone has certain pathologies of either the kidneys or of the blood, etc. Okay? Here's an individual doing the dipstick test. So they dip this into the urine sample and then read in the direction with what are each one of these. This is a different test from this, and this. So what I'm saying to you is that each of the little pads there is a specific biochemical test for the urine. And if you do it right and you time yourself right, you're going to get a lot of information very quickly. And a lot of times this is done right in the office, as you can see here. And so these numbers and these colors will tell you exactly what is the condition for basically all sorts of things, pH, sugar, and a variety of other tests. And you can see this here. You will use a urine color, you will have specific gravity, you will have noticing if there are any unusual characteristics of tone. You will be able to detect for ketones, bilirubin, glucose, protein, urobilinogen, nitrile, leukocytes, etc. This is just fantastic. Okay, now review the gross anatomy of the urinary system, including any models and or dissection model. And in this case, go over the models that you see in the text, as well as the models that you will see in some of the videos, etc. Review the microanatomy of the nephron, the renal blood supply, ureters, and bladder, including review the slides and the micrographs. Review and examine the physical and chemical characteristics of urine, relate the function to the nephron structure. In the lab, we are not doing the actual urine analysis. You should review and view the lab videos that cover urinalysis, because these are going to be absolutely essential. Then fill out the worksheet, which includes pages 535 to 536, 547, 548. Turn in the worksheets. Upload them by 5 p.m. this Friday. Okay? And have a wonderful day.